Hello, class. This video is going to look into a topic that biologists frequently refer to as tree thinking. And tree thinking basically refers to our understanding of the evolutionary branching patterns of life and how we sort of have evaluated the relatedness of different living things, different lineages that have existed, particularly those that exist currently, but also some that have gone extinct in the past. And this also leads us to thinking a little bit about the process of classification. And so that is the means by which we make determinations about how different groups of organisms are classified based on certain characteristics that they share with one another, but don't share with other groups. And that leads us to a field called taxonomy. And so we will begin by kind of talking about taxonomy. Well, what is taxonomy? Taxonomy is, by definition, a system of naming and classifying organisms. And people have been doing taxonomy for a very long time. So, for example, we might look at a whole diversity of different kinds of woodpeckers. And ornithologists have figured out different ways to name these things based on certain features they have, but also classify them according to certain characteristics that some woodpeckers share with other woodpeckers that are more similar to characteristics that other woodpeckers have, for example. And taxonomic classification usually is based on shared characters, right? So if two different organisms share a certain characteristic in common, we would consider them perhaps to be more closely related to one another than a third organism that didn't share that characteristic in common. And these characteristics could be what we call morphological traits. These are physical features. And in fact, most of the earliest work in taxonomy was based on using physical features to classify things. But also, more and more nowadays is being based on genomic information, or the information contained within DNA, the information contained within genes. And sometimes we see that the information contained within genes agrees with classifying things according to their physical features, and sometimes maybe it doesn't so well. We'll talk about that in a moment. This is sort of our modern understanding of taxonomy, but I think it's important to point out the fact that taxonomy really does predate science, and this falls under the umbrella of something we would call folk taxonomy. And folk taxonomy really represents the cultural origins and the cultural history of naming different living things. All kinds of different cultural groups around the globe have recognized that there is diversity in life, and all kinds of different cultures around the globe have come up with different systems for naming things and different names for things. And in fact, although we use certain very specific scientific names nowadays, and certain scientific classification schemes, all of that is predated by folk taxonomic systems, folk taxonomic naming systems. For example, here in California, we have a handful of different indigenous groups here, the Chumash, the Cahuilla, and the Kumeyaay. And anthropologists have done work with some of the surviving members of these groups who have retained some knowledge of their folk taxonomy and published books, in particular books on what we call their ethnobotany. And so Jan Timbrook published this book on Chumash ethnobotany. So this book describes a whole multitude of different native plant species in California, but it describes the names that the Chumash used for these plants. And not only the names that the Chumash used for these plants, but the way that the Chumash used the plants to do stuff. And a lot of times in folk taxonomy, the naming system of different organisms was associated to how they could be useful. This is also true of this book, Temopak, which was published by Catherine Silva-Sabel, who was a Cahuilla elder since passed, and also a similar book published on the ethnobotany of the Kumeyaay people who occur more down in San Diego County and down into Baja California. But the idea is that, to emphasize, that the system of naming things is predated by a more Aboriginal perspective compared to that which we now think of as being quite more scientific. And the sort of modern origin of scientific taxonomy really begins with Carl Linnaeus, who had proposed this hierarchical rank-based taxonomic classification system, which we still use today. And you've probably heard species names used before. And so Linnaeus is the one who came up with this idea of naming species. So we might think of species or organisms as having common names like the American black bear, but that American black bear also has a scientific name, which is Ursus americanus. And one of the important differences between the scientific name and the common name is that the scientific name really applies to people who speak any language other than English. So American black bear, of course, is an English description, but scientists anywhere in the world can see Ursus americanus and know that that is the organism that we are talking about. And of course, this classification scheme is pretty famous for nesting smaller taxonomic groups within larger taxonomic groups. So things like the American black bear, which is a species, belongs to a genus, and that genus is Ursus. And the genus Ursus belongs to a family, Ursidae. This family belongs to an order, Carnivora. This order belongs to a class, Mammalia. This class belongs to a phylum, Chordata, which are the vertebrates. And this phylum belongs to the kingdom Animalia, et cetera, et cetera. This classification system was pretty widely accepted at the time because it is hierarchical. And it, again, was fairly aligned with thinking of early Greek philosophers, that there was this hierarchical nature in the distribution of biodiversity, which we know now is actually not true. Although the system was originally designed with thinking that species didn't evolve and thinking that species never changed from being one species to another, we still are applying this today, even though we know that that case is not true. Species do evolve and species do change. And so by virtue of that fact, this classification system gets reorganized all the time. These schemes are subject to what we might call taxonomic revisions pretty frequently, where we say, oh, the relationships that we once thought were correct amongst a certain group of organisms might not be correct because we have new evidence to suggest that different groups are more closely related than we had previously thought or not, based on better morphological data, or nowadays more and more based on genomic data, that is data from DNA. Anyway, this is sort of the basis of thinking about classification and the basis of what we call taxonomy. This leads us into a kind of more specific branch of taxonomy that is widely utilized within the study of evolutionary biology today, and that is something called phylogenetic systematics. And phylogenetic systematics is effectively the study of classification and evolutionary relationships among organisms. So we are still trying to classify things based on their relatedness, but now we're saying we specifically want to do that in the context of how things share common ancestry. And this idea has led to the concept of building something called a phylogeny, which is sort of like a tree. If you can look at this cartoon here, these sketches are sort of postulating that life has evolved through some tree-like branching patterns. And this image here actually is the very first phylogeny ever proposed, although it's entirely hypothetical. It is not intended to include any actual living organisms. It's only intended to illustrate the concept that there is this branching pattern. And this is actually the only image that was published in Charles Darwin's famous book on the origin of species. And this branching pattern that he's illustrating here is really much trying to demonstrate this concept that he first presented in this book of descent with modification. So Darwin's kind of saying here that all organisms in this particular tree share a common ancestor at some point in time, which is here. And as we move from the bottom of the page to the top of the page, we're going from the distant past to the more recent future. And we can see that lineages are beginning to branch, and they're branching off from a common ancestor. And then that leads to other common ancestors that themselves start to branch off. And there's just tremendous amount of different branching that's going on that Darwin proposed had led to the ultimate diversification of life as he knew it at the time, and as we know of it today. Well, as it turns out, interpreting these branching patterns have become extremely useful in understanding taxonomic relationships. And the field of phylogenetic systematics has shed tremendous amounts of light on the process of shared ancestry via the process of evolution. So two processes that kind of lead systematic evolution of higher taxa that we do know of now, that Darwin hadn't really discussed, but he came up with some pretty important inferences to support, are the ideas of anagenesis and cladogenesis. And basically, the idea is if we have this tree here, this is or a piece of a phylogenetic tree here, we can think of traits evolving along this tree it in some way from some perhaps lower value to say, let's say smaller to some higher value. Let's say things are getting larger. And we can sort of interpret how traits are changing over time. And basically, we could think of this tree as being composed of a sort of ancestral lineage we'll call A, and in some point in time, the ancestral lineage called A, which is sort of evolving to become larger and larger, branches off into two subsequent lineages, B and C. And maybe the lineage B actually started to evolve to become smaller, kind of more like the ancestral state, whereas the other descendant lineage C starts to evolve to become even more and more larger. And as time progresses, C evolves into D, and the evolution of C into D marks a significant change in, let's just say, size, trait value, going from moderately sized to becoming much larger. And this process is what we call anagenesis. It's evolutionary change within a single lineage. But then later on down the road, this lineage branches off into two subsequent lineages, E and F. And E, the lineage giving rise to E, kind of starts to revert back to the more ancestral state by becoming smaller, let's say, whereas the lineage giving rise to F is continuing to evolve to become larger and larger. And that process of diversification is called cladogenesis. That's branching of a single lineage into two descendant lineages. So we have these two kinds of different ways that we can think of lineages within these branching trees changing. Now, this example is entirely hypothetical. Trait values don't always just evolve from small to large. It's much more complicated than that. But for the sake of conversation and simplicity, this is kind of a good way for us to illustrate these two important processes that explain systematic evolution. So again, to reiterate, basically this science or this subfield of evolutionary biology, which we call systematics, is basically striving to classify taxonomic groups that can convey evolutionary relationships using these phylogenetic trees. And phylogenies basically look something like this. They are trying to infer patterns of shared ancestry among related lineages. And so if we take 4 different types of organisms as an example, we can plot them onto our phylogenetic tree. And these organisms are generally referred to as taxa. And taxa could be species, taxa could be different genera, taxa could even be different families. So some phylogenetic trees show species, some phylogenetic trees might show genera. In this case, we're looking at really broad groups of organisms. Bacteria, well, that's a really diverse taxonomic group, but we're going to say all bacteria are included here. Birds is also a pretty large taxonomic group, so we're going to say all birds are included here. Marsupials, again, a pretty diverse branch of the mammalian lineage. We'll say all marsupials are included here. And then the genus Homo. Well, okay, the genus Homo only includes one lineage that's still alive today, but once upon a time, there were other different species of Homo that existed. And so each of these different groups of organisms are referred to as taxa, which is a plural form of the word taxon. And so we could say that bacteria on this tree represent a taxon. Birds likewise represent a taxon. Marsupials represent a taxon, and likewise Homo. And an important piece of terminology that often comes up is that the most closely related lineages that occur on these trees are generally referred to as sister taxa. So for example, marsupials and the genus Homo would be considered sister taxa on this tree because they share an awful lot of evolutionary change in common. For example, they are both mammals, or two lineages comprised of mammals. Now, these phylogenetic trees usually contain something called an outgroup, And so that's basically the earliest diverging lineage that we see in the tree, and that's frequently referred to as a basal taxon. And oftentimes these trees will have a root. And so the root of the tree really represents the common ancestor of all the different taxa on the tree. And basically, these trees infer time, kind of as we saw in the Darwinian image, going from a very, very distant common ancestor of all things to a more recent descendant taxa. And so, likewise, we're seeing that on this tree, where we're going from time in the very distant past to time in the present. And these trees also contain these things called nodes. And nodes, very importantly, represent common ancestors. So this tree has 3 nodes. This node represents the common ancestor of all of the descendant taxa on the tree. This node represents the descendant ancestor of basically vertebrates, that's birds, marsupials, and Homo. And this node here represents the common ancestor of the mammals. And these nodes are not dissimilar from these nodes that we see representing events of cladogenesis, some type of really important evolutionary change occurring at these points in time by which lineages diverged. And so all of the evolutionary change that occurred from the time of the common ancestor of all taxa to the divergence of bacteria and vertebrates is shared by all the lineages on the tree, but the evolutionary change that occurs after this point in time is only going to be shared by the vertebrates and not by the bacteria. Likewise, all of the evolutionary change that occurs after birds split from other vertebrates is going to be shared by all these lineages. Let me rephrase that. All the evolutionary change that occurs after these birds split from the other vertebrates is either going to be shared by the birds independently or by the other vertebrates independently. So the evolutionary change that is starting to occur at this point in time is not shared between these mammals and these birds. And likewise, there are certain differences between marsupials and the genus Homo. At this particular node here, marsupials have gone on to undergo different types of evolutionary change than have the lineages giving rise to Homo, and that is that these marsupials are monotremes, and these Homo sapien, or Homo, the genus Homo, are placental mammals. So this is a basic rundown of how you might interpret a phylogenetic tree, a very simple example of one, albeit not a great phylogenetic tree. Usually we actually like to see a little bit more information on a phylogenetic tree, and we'll talk about that in a moment. But when you're looking at these trees, this is kind of how you want to think about what the information they're showing actually represents. Now, these phylogenetic trees, as it turns out, can be depicted in a whole variety of ways, which makes them kind of confusing. And so, in order to sort of negate that confusion, I invite you to realize the fact that all phylogenies are really trying to show the same kind of information. They're trying to illustrate this pattern of descent with modification, or these various stages of things like cladogenesis and anagenesis. So despite the fact that we can have a variety of different ways these trees are drawn, they're all depicting the same kinds of information. So we can have rooted trees, sort of like the one we just saw, and those rooted trees can be oriented in different ways. For example, we can have a tree that's shown going in a linear fashion, from left to right, that is. And so now in this case, time is going from the distant past to, say, the present in this particular tree. This tree here is the same tree as this tree here. It's just been flipped 90 degrees. And so once again, here we have time in the distant past and going to time in the present. And this tree here is the same tree as this tree here. The only difference is that the artist of the tree decided to include little elbows on this tree and this tree, but they did not include little elbows on this tree. The little elbows are 100% completely meaningless. They're only for sake of illustration, but they don't infer any meaningful information about the actual evolutionary relationships shown in the tree. So as it turns out, these 3 trees are identical. They're showing identical patterns of shared ancestry amongst these 5 taxa, A, B, C, and E. This is an unrooted tree here, so it doesn't have any suggestion necessarily about a distant common ancestor, and the authors of the tree may or may not have had reasons why they didn't need to show that, but it's a little bit crazier looking. but it's also based on some crazier type of data that was used to construct the tree. But it's still inferring that there is some kind of a common ancestor that these things share, and there are a whole bunch of descendants. There are cartoon trees, so sketch trees that people just might draw by hand to sort of uh, illustrate some inferences that they might have about different patterns of evolutionary relationships, but maybe they're really rudimentary in nature. This particular tree here was a sketch drawn by Charles Darwin. And this was Darwin trying to, again, illustrate his concept of descent with modification and this idea that probably life has evolved via these various patterns of lineages branching off from one another, but they all share a common ancestor. And then we can have these circular trees, which are rooted trees. And the reason why people build circular trees is because sometimes there's so much information to be included within the tree that it just doesn't fit very nice into, say, one of these types of scenarios or one of these types of configurations. And so this tree here is the tree of life, of all life. And it starts all the way down here with the last universal common ancestor. And the earliest lineages that branch off are the bacteria and archaea. Only later do the protists branch off, the so-called protozoans. Only later than that do we see the lineages giving rise to plants and animals and fungi branching off. And of course, our own species is included within this tree, albeit it's very difficult to see how we fit in because there's so much information on the tree. But it's not uncommon to occasionally see circular trees when there are a lot of taxa being included on the tree. Again, the different orientation of these branches, so or different configurations of these trees don't change the information that's being presented. So if you were to consider these three different phylogenetic trees here and these three different phylogenetic trees here, guess what? All six of these trees are showing the same patterns of relatedness. The trees shown in sort of column A don't have these elbows, whereas the trees shown in column B do have these elbows, but the elbows don't convey any important information at all. One sort of important difference that you might notice is that if we compare this tree to this tree, we see that taxa 1, 2, and 3 are sort of arranged in numerical order, whereas in this tree, they're not. It's sort of going backwards. But that doesn't change the information being presented. And this is kind of like saying that reorienting sister taxa in a tree is analogous to spinning linked branches in a mobile. You might be familiar with these mobiles. These mobiles have these things that come down, and they have these nodes that connect to other things. And these things can spin around each other. And whether or not these things are oriented in one way or oriented in another 180 degrees against that way, the relationship they have with that node is not changed. And so this is kind of like saying there's a relationship between my thumb and my fingers. And if I flip my hand around like that, that doesn't change the relationship between my thumb and my fingers. You're just looking at my hand in a different way. Likewise, I have two hands, and I can orient this hand around this way. That doesn't change the relationship between my arms. I can orient my hand back this way and the other hand this way. That doesn't change the orientation or the relationship of my hands to my body because my hands are tied to my arms, which are rooted to my body, which is kind of like the node. And you can think about my wrists as also being nodes. And so it's important to recognize that in the variety of ways that these trees are depicted, they're really always just trying to show evolutionary relationships, and they can be drawn in a multitude of ways that show the same information. And so when we talk about the most important feature of these trees really being an illustration of patterns of shared ancestry, we are talking about shared traits. And very much so, these traits are constructed, these trees are constructed using shared traits. And the shared traits most often used to construct these kinds of trees are a special kind of shared traits we call homologous traits. And a homology is specifically a shared character that's been inherited from a common ancestor. And these shared characters could be derived traits, that is to say that they've evolved more recently, or they could be ancestral traits, that is to say traits that evolved a long, long time ago that a whole bunch of descendant lineages still have. So for example, if we look at this particular phylogenetic tree here of different vertebrate forms, different vertebrate organisms, we have 5 different mammal taxa on the tree: rats, beavers, humans, and kangaroos. Sorry, 4 different mammal taxa. And these different mammal taxa share some different traits in common. Specifically, the 3 traits that make all 4 of these taxa mammals are the single jawbone, the fact that they produce milk, and the fact that they all have hair. And so these are homologous traits that are also ancestral traits amongst all mammals. We can talk about some more recently evolved traits as being more like derived traits. So things like the placenta in the lineages giving rise to rats, beavers, and humans. Kangaroos don't have a placenta. So the placenta is a more recently derived trait in a certain group of mammals. Likewise, the ever and growing sizers of rodents are derived traits that only rodents have. Beavers don't have these traits, and of course, humans don't. And so when we're looking at a lot of phylogenetic trees, we are looking at trees that were constructed using homologous traits. However, as it turns out, not all traits are inherited from a common ancestor. That is, not all shared traits. For example, bipedalism in certain vertebrates, or certain organisms that might have the same color. Well, they didn't necessarily inherit that color from a shared ancestor. Or things like flight is a very classic example of a trait that has evolved independently on multiple occasions. We'll discuss that in a moment. But so, for example, like bipedalism, right? Humans walk on 2 legs, but so do birds. But did humans and birds inherit bipedalism from a common ancestor? The best evidence points toward the answer being no, they did not. Humans evolved bipedalism independently after they branched off from their other ape-like ancestors. And likewise, birds evolved bipedalism after they branched off from some of their reptilian ancestors. And so sometimes traits that are really similar that different taxa share are not inherited from a common ancestor, and therefore they are not homologous. They are instead what we call analogous traits, and they arise via a very special process that we call convergence, and I'll explain how that works in a moment. A very important tool used in resolving or sort of trying to understand different evolutionary relationships comes from the field of comparative anatomy. And comparative anatomy uses homology to find the most likely patterns of shared ancestry among different taxonomic groups. A classic example of this comes with the homologous state of tetrapod limbs. And so all tetrapods inherited their limb form from a common ancestor that existed actually a very long time ago. And that might have been something that was very similar to a salamander, something that was sort of semi-aquatic and semi-terrestrial, such as the classic famous fossil Tiktaalik. But we know that all tetrapods share their limb forms in common. However, these limb forms have been significantly modified. And this is kind of not too different than Darwin's idea of descent with modification. So we can think of the earliest tetrapod limb as looking something like this. It had a humerus, it had a radius, an ulna, and carpals, and some metacarpals and some phalanges. And we see that some descendant lineages of tetrapods, like seals and horses and humans, have a very, very similar morphology in their limbs. However, these morphologies have been highly modified, and they've been highly modified because natural selection has favored certain variations that allow these different modifications to perform a whole multitude of different functions. For example, the limbs of seals are adapted for swimming. And so this has favored a larger humerus and a larger ulna, and maybe slightly longer carpals and metacarpals and phalanges. But the same structures still remain. They are homologous structures. Likewise, in humans, we see that maybe these structures have been favored for grasping rather than they have been for swimming. And so we still see these similar features in humans, but in this case, the humerus has become elongated, and the radius and ulna likewise have become elongated. We can look at the limbs of horses. which are also homologous to these limbs. But in this case, the carpals, metacarpals, and phalanges have sort of become fused together, really favoring what we might call phalange number 3. And so this particular structure of the horse is something that has given it tremendous abilities to be adapted for running. But the overall sort of plan, the overall body plan here is still the same plan. We see even more sort of extreme modification of these forms in things that fly, flying tetrapods. So for example, bats. Well, bats are mammals, and bats obviously can fly, and they still have this quintessential body plan that they inherited from their tetrapod ancestor. Of course, the carpals, metacarpals, and phalanges have been highly modified in the wing structure of bats, but nonetheless, they are homologous to fingers in humans, or even similar type structures in seals that we can't so obviously see as fingers. In birds, there's been even more sort of extreme modification of this form. Birds have the humerus, radius, and ulna, but they have a reduced sort of set of fingers. They only have 3 fingers in their wings. So 2 of those fingers have either fused to the others or have been lost altogether. And then even things like pterosaurs, which were these ancient flying reptiles, have a very, very similar body plan, albeit they did have an extra sort of special bone called a sesamoid that is not seen in these other vertebrate examples. But the body plan still is very much the same. There is a humerus, there is a radius, there's an ulna, and you have this really, really elongated 4th phalange. So all of these characteristics have enabled evolutionary biologists to start to make inferences about the fact that seals, horses, humans, bats, birds, and pterosaurs all share a common ancestor at some point in time that was some type of an early tetrapod. And from this common ancestor, they inherited all of these characteristics. And these characteristics have simply been modified because these different organisms have evolved in a multitude of different environments. And so there are environments where adaptations for swimming are more valuable than adaptations for running compared to adaptations for grasping or adaptations for flight. Developmental biologists have also used homologies to start figuring out relatedness amongst different taxonomic groups. And we call these developmental homologies. And so the way that organisms develop from their earliest inception, that is from the time that sperm met egg via fertilization, at least in these sexually reproducing vertebrates in this example, the way that organisms begin to develop from that earliest stage of fertilization has some shared characteristics in common and some characteristics that are very different amongst different lineages. So we can look at humans, monkeys, pigs, chickens, and salamanders. And following fertilization, their zygotes are all very, very similar physically. Following that, the earliest stages of development from there, they start to form some kind of similar structures as cell division unfolds. But then we can see that as these really early developing blastophores start to turn into these embryos, or look more like embryos, there are some pretty succinct differences that we can see between these things. Obviously, humans and monkeys have embryos that are a lot more similar to one another than they are to those of salamanders. Likewise, we can see that maybe salamanders and chickens have some similarities that are a little bit more common than they are to these other forms of mammals. But basically, this sort of pattern of what we call ontogeny or early development points towards patterns of shared ancestry, and we can use these patterns to build a phylogeny. And so we might be able to say, the shared characteristic of the way that these early zygotes are unfolding points toward the most common recent ancestor of terrestrial vertebrates being an organism that had that characteristics. Only later on, the sort of differences we see in the blastophores between these mammals and these other vertebrates are probably those that diverged when those different organisms shared their common ancestor. And likewise, the most recent common ancestor of all the mammals is giving rise to some of these characteristics, so on and so forth. So not only actual anatomical traits that we observe in sort of mature life forms can point towards their shared relatedness or patterns of shared ancestry based on homologous traits, but we can also look at the way that things develop in their earliest stages, and that can give us some clues as well. Now, there's this other kind of trait we sort of talked about that's an analogous trait. Those are shared traits among taxa that are not inherited from a common ancestor. And these traits are referred to as homoplasies. So homoplasies are kind of in contrast to homologies. And again, a classic example of homoplasy is flight. So for example, we know that among certain insects, there are wings. And these wings, of course, enable them to be able to fly. So things like the Lepidoptera and the Diptera, which are flies, Lepidopteras are like moths and butterflies. These things inherited wings from a common ancestor. So wings amongst these two lineages of insects are homologous. However, that's not true of wings in chordates and wings in arthropods in general. So for example, hummingbirds are vertebrates, and hummingbirds can fly. In fact, many, many different species of birds can fly, and many, many different species of arthropods can fly. But these two different lineages are separated by an awful lot of different evolutionary change. And as it turns out, these hummingbirds or these chordates have sister taxa, echinoderms, that certainly do not fly at all, that they are a lot more closely related to than they are to arthropods, which of course fly as well. And arthropods have other taxa that are more closely related to them than chordates as well. Of course, they all share common ancestry at some point, but what we're trying to get at here is the fact that did flight emerge originally way, way back when, only to be lost on multiple occasions, or does it make more sense to say that flight evolved, or rather wings evolved independently on multiple occasions? And the best evidence points towards the latter explanation, that these wings did evolve on multiple times on more than one occasion. And that's why it is what we see distantly related taxonomic groups sharing certain traits. And this happens generally probably because these traits were being favored under very similar environmental pressures in very similar different environments. So natural selection was operating on these kinds of traits in similar ways in different environments, even though the different lineages that selection was operating on were pretty distantly related. And these analogous traits can be favored in something we might call different or comparable ecological niches. So really similar types of environments or really similar types of niches can favor really similar types of traits being favored by selection, again, even though things are not inheriting these traits from a common ancestor. A classic example of this comes with succulent plants. So we can look at these three pictures of different succulent plants here, and you might be inclined to say, well, these are all cactus, right? But actually, they're not all cactus. The only one that is a true cactus is the one in the middle, which belongs to the genus Echinocereus. This example on the left is a euphorbia, and this example on the right is a hoodia. And these different succulents are in three completely different plant families that are very quite distantly related. Succulence and spininess was not inherited from a common ancestor in this case. Those two traits arose independently on multiple occasions in different parts of the world, as you might notice here. And that's because the different environments where these different succulent and spiny organisms evolved were under very, very similar environmental pressures, probably areas that were very dry, favoring succulents, the ability to retain water, probably in areas where these plants were susceptible to herbivory, susceptible to being eaten. So they evolved spines to prevent them from being eaten. Another classic example comes from comparisons of placental mammals and Australian marsupials. And here we have a whole bunch of very similar forms that occupy very similar types of ecological niches. And again, these different niches can refer to a sort of set or let's just say a set of different aspects of the environment that organisms occupy. Things like the type of climate they prefer, the type of habitat they prefer, or the type of role that they perform in an ecosystem. These types of niches exist in different parts of the world, and different organisms have evolved into them. So in the case of these two contrasting types of mammals, we have burrowers. And of the placental mammals, we have moles that burrow. But of the Australian marsupials, we have marsupial moles. We have placental anteaters, but then we also have marsupial anteaters. We have both placental and marsupial mice take on a very similar type of morphology, but they didn't inherit it from a common ancestor. Likewise, things that climb, we have placental lemurs, but then we have the spotted cuscus, which is a marsupial. Flying squirrels that are placental, or flying phalangers that are marsupials, et cetera, et cetera. We see examples of different kinds of mammals with really, really similar morphological traits, really, really similar ecological niches, but these traits did not come from a common ancestor. They are homoplasies. And so we might be asking how these homoplasies arise. They basically are either going to arise via something called convergent evolution, which again is this process of very similar environmental pressures favoring really, really similar traits in distantly related groups, or they could also occur via something we call evolutionary reversals. And so a couple more examples of this. We can look at these flowering plants here. And basically, the flowering plants on the top row here all have what we might call bilateral symmetry in their flowers, whereas these flowering plants on the bottom all have radial symmetry in their flowers. And so these flowers are sort of radially asymmetrical, and these flowers are sort of bilaterally symmetrical. And you might be inclined to say, well, the ones on the top have to be each other's closest relatives, and the ones on the bottom have to be each other's closest relatives because they share these similar features. But actually, if we instead look at this image in terms of its columns, it turns out that these things, which are called monocots, are each other's closest relatives. These things, which are called eudicots, are all each other's closest relatives. And within these eudicots, we can look at fabids that are each other's closest relatives, and things in an order called the Malpigales, and they are each other's closest relative. And so within different related lineages, bilateral forms of flowers and radial forms of flowers have evolved independently on multiple occasions. And this is a classic example of convergent evolution. Certain situations where having radial flowers was more favorable than having, radial flowers was more favorable than having bilateral flowers, et cetera, et cetera, et cetera. This other story of evolutionary reversal is basically the scenario where a trait evolved and it was conserved for a long time, but then in more than one lineage, that trait became lost. One or more lineages, that trait became lost. And a good example of this is wings in insects. So these are basic. This is basically a phylogeny of major insect groups. And the ancestral forms of insects did not have wings. And so some of the early diverging insects, such as bristletails and silverfishes, they don't have wings, they can't fly. But then all of a sudden, wings originate, and many of the lineages following thereafter, such as dragonflies and crickets and bugs and flies, have wings. However, we see some taxa amongst the higher lineages of insects that have lost their wings. There are wingless crickets. There are lice, which are actually closely related to bugs, but these lice lost their wings. There are fleas, which are pretty closely related to flies, but these fleas lost their wings. So on 3 separate occasions, we see this trait of wings being lost in these different lineages of insects. And you might be inclined to say, well, fleas, lice, crickets, silverfishes, and bristletails must all be each other's closest relatives because they don't have wings. But more robust advanced analysis has pointed us towards the fact that that's not true. And the fact is that on multiple occasions, these traits have been lost. And so these are examples of homoplasia. And so just to reiterate, these homoplasies are traits shared amongst taxa, but traits that these taxa did not inherit from a common ancestor. So for example, again, the bilateral floral symmetry amongst these particular taxa, not inherited from a common ancestor. The radial floral symmetry amongst these taxa, not inherited from a common ancestor. The state of winglessness in these certain insects, not inherited from a common ancestor. So we'd like to look into a little bit of detail about how it is that these trees are constructed using actual data. And classically, this has been done using something called character mapping. And so we might have a table such as the one shown here that includes some taxa. So we have mosses, ferns, pines, and oaks. These are all plants. And we have some traits that these taxa may or may not have. And if the taxa have the trait, the table shows a little plus sign. If the taxa don't have the trait, the table shows a little minus sign. And this can allow us to construct an evolutionary tree with some amount of accuracy. So for example, if we only looked at a couple of these traits, such as the fact that we know all these things carry an embryo because they're all eukaryotic and they all undergo sexual reproduction, we can use that as a most basic trait at the root of the tree. But then later on, we have this trait of xylem and phloem. Well, it turns out that mosses don't have that trait, so that enables us to make an educated guess about them being the outgroup or the earliest diverging lineage in this tree. But then the relationship amongst ferns, pines, and oaks becomes unresolved if the only data we have is the fact that they all carry xylem and phloem. If we start to account for the fact that some of these things have wood and some of them do not, some of them carry seeds and some of them do not, and some of them produce flowers and some of them do not, then we can make a much more accurate tree. For example, we know that ferns, pines, and oaks all have xylem and phloem, and so that is a shared trait amongst these taxa, a homologous trait. However, ferns don't have wood and don't have seeds and don't have flowers, so that kind of sets them apart from pines and oaks. And then pines and oaks, of course, both do produce wood and produce seeds, but oaks produce flowers and pines do not. And so that kind of teases them apart in this phylogeny. And we're able to piece together their evolutionary history based on really just a basic amount of information. And so this type of an approach kind of gets at building trees based on groups that are sharing features in common that are making them appear to be more closely related to each other than to groups that don't share those features in common. And this leads us to a process by which evolutionary relationships are pretty frequently inferred, and that is by something using the principle of parsimony. And parsimony is kind of just a fancy word for saying the tree that implies the least amount of evolutionary change is the one that is most likely to be correct. And so let's consider three possible trees that each have three different taxa and that uses these each of these trees only use two different characters to try to construct the tree. Well, if we started to play around with this, we'd quickly come to the conclusion that there are three possible ways that these taxa could be related. And these taxa in this case are lizards, humans, and dogs. They're all vertebrates, thus they all have a backbone, but only humans and dogs have hair. And so if we were to suggest that perhaps, okay, the backbone is the ancestral trait that all taxa share, it's homologous, that's kind of inferred in each of these trees. But then there are a couple different ways that humans and dogs could have inherited hair. They could have inherited from a common ancestor, or hair could have arisen independently in the lineage giving rise to humans, only later arising independently in the lineage giving rise to dogs, suggesting that dogs and lizards are each other's closest relatives, or the opposite scenario, that hair gave rise to the lineage of dogs, only later to have arisen again in the lineage of humans, suggesting that humans and lizards are each other's closest ancestors. Now, of these three trees, which one involves the fewest number of evolutionary changes? The first tree does. It only involves two evolutionary changes. The backbone evolved once and hair evolved once. This is the most parsimonious tree. It makes less sense to believe that hair could have evolved on multiple occasions because it takes a lot for these different traits to evolve on more than one occasion. Now, we know that does happen sometimes, obviously, because there are homoplasies, and these homoplasies occur because of convergent evolution or reversals, which are instances where, yeah, maybe certain evolutionary events did occur on more than one occasion. Maybe wings were lost in insects more than once. Maybe bilateral floral symmetry evolved more than once. Maybe radial floral symmetry evolved more than once. But by and large, the most fundamental concept used to construct a phylogenetic tree is using the principle of parsimony to take, again, an educated guess that the fewest number of evolutionary changes that had occurred in the past are most likely to be accurate. We can also build parsimonious phylogenetic trees using DNA sequence information. And in fact, that is very much more the way more so the way that folks are doing this today. So we can look at these 3 different trees that are composed of basically 5 different species each. And what we do know about the species is that they carry a certain sequence of DNA at a certain region of the genome. And we're trying to make inferences about how it is they got to the point of having that particular sequence of DNA. If we make the assumption that the common ancestor of all the taxa had this particular sequence, AAA, 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 there are a few different ways we might get explaining how it is we got to the descendant taxa carrying sequences like TTA or TTG or AAG or AAA. And there's a couple different avenues by which we can get to these conclusions. And basically, we can look at these trees at face value and see that this tree involves 4 different evolutionary changes leading to these taxa carrying those particular gene sequences. This tree involves 5 different evolutionary changes, and this tree involves 6 different evolutionary changes. So at face value, just considering the fact that this tree here, tree A, that involves only 4 different periods of evolutionary change from a common ancestor to these descendant taxa, is probably the one that's going to be most accurate. And so again, these types of techniques are very much those that are used by evolutionary biologists to reconstruct evolutionary relationships. Phylogenies generated with molecular evidence, such as the characters we were looking at here, such as xylem and wood and seeds and flowers, etc. Trees generated with, this is molecular evidence. Phylogenetic trees generated with molecular evidence corroborate well with those using phenotypic evidence, this being the phenotypic evidence, this being the molecular evidence. And comparing the methodologies confirms much relatedness amongst these different lineages using independent data sources, multiple lines of evidence. We can take morphological data and we can take genomic data and use it to build a phylogeny of these various types of animals. And as it turns out, these 2 phylogenies are remarkably similar. And this suggests that both approaches work pretty well to point towards giving us a pretty similar answer about what it is we understand about the evolutionary history of different. Now, phylogenetic trees are often depicted according to the information used to construct them. And so there are kind of like 3 different types of phylogenetic trees in terms of the information being used to construct them. We have cladograms, phylograms, chronograms. Basically, the main difference between them is the lengths of the branches. A cladogram or cladogram is kind of the simplest form of a phylogeny. The lengths of the branches don't mean anything. These cladograms show general assumptions just based on morphological comparisons or maybe fossil data, but they don't make any inferences about the actual amount of shared genetic information or the actual amount of time that had elapsed since these things shared common ancestry. It's simply just an illustration of the fact that they share ancestry at this point, they share ancestry at this point, et cetera, et cetera. For example, these trees here are kind of like cladograms. This is in contrast to something like a phylogram, which is taking DNA data to really illustrate relative amounts of genetic change amongst taxa. And so certain branch lengths here are representing how much genetic change has elapsed since these various taxa last shared a common ancestor. So for the most simple example, we could say, well, this is the common ancestor of all the taxa on the tree. It diverged into 2 subsequent lineages, and 5 genetic changes happened, leading to the present day state of taxon D, and 3 genetic changes arose, giving rise to the common ancestor of the other 3 taxa. And likewise, maybe only 1 genetic change gave rise to taxon A, and 1 genetic change gave rise to the common ancestor of taxons B and C. Likewise, 1 genetic change then gave rise to taxon C, and 6 different genetic changes gave rise to taxon B. So in this particular scenario, we're saying the lengths of the branches as illustrated in the tree represent relative amounts of genetic change. A chronogram, also known as an ultrameric tree, but a chronogram very similarly is showing estimated divergence times amongst these lineages. So in this case, the branch lengths don't represent amounts of genetic change. The branch lengths represent time. And so the amount of time that has elapsed from the common ancestor of all the taxa on the tree to the emergence of taxon D is much longer than the amount of time that elapsed since the emergence of the common ancestor of taxa A, B, and C. Likewise, the amount of time that elapsed between the emergence of taxon A from its common ancestor of taxa C and B was pretty short. And so in this idea, we're getting a better sense of how long has it taken for evolution to happen. Whereas here, we're getting an idea of how much genetic information do these things share. And here, we're not really getting any more information other than which taxa share their most recent common ancestors. So then we can sort of start talking about the types of classification schemes that evolutionary biologists use based on this type of information, based on patterns of relatedness that are revealed through building phylogenetic trees. And different taxonomic units are usually grouped according to these patterns of shared ancestry. And these classification schemes come in basically three different forms, different ways that we can group the taxa and ancestors that are shown within these trees. One way, which is probably the one you hear the most about, is to group things into what we call monophyletic groups or clades. Another way is to group things into something called polyphyletic groups, and yet another is to group things into something we call paraphyletic groups. Now, this nomenclature is a little bit, if it's a little bit uncomfortable for you, don't worry about it. That's not too much of a big deal. The important thing is to realize that these grouping systems look a little bit different, and I'll walk you through how that works. So when we talk about monophyletic groups, we're talking about groups that include a common ancestor and all of its descendants. And that is nicely referred to as a clade. A classic example of clades, of a clade, can be found in plants. And so we can look at angiosperms, which are basically flowering plants, different types of conifers, or what we might call gymnosperms. But we can look at these different lineages of plants. And at this particular point in time, all of these groups share a common ancestor. And this group as a whole shares all of the descendants from that common ancestor. And this particular group is one we call seed plants. All of these things inherited the characteristic of producing seeds from their most recent common ancestor. And so that forms a clade. Likewise, we could say that this group here, which does not produce flowers, but does pretty much everything else that flowering plants do, form a clade as well, because they share a common ancestor at this point in time, and all of the descendants of that common ancestor. So these are what we call clades. These things called paraphyletic groups are basically groups that include a common ancestor and some, but not all of the descendants. And this is kind of a confusing classification scheme, but it basically has occurred because of a lack of knowledge about the relatedness amongst organisms. One of the most classic examples of this comes from the relationship between reptiles and birds. Formerly, the group of reptiles was called Reptilia, and Reptilia inherently formed a paraphyletic group because it included a common ancestor shared by all the reptiles, but it also includes these bird descendants that are not included within the reptile group. And so by virtue of this fact, we see a group where, again, they share common ancestry, sort of as we see here, sort of as we see here, and they share some of the descendants, such as snakes and crocodiles. But as it turns out, crocodiles actually are a lot more closely related to birds than they are to snakes. And so this causes kind of a mess in the classification scheme. Folks have recognized this mess and figured out ways to resolve the situation. So by instead classifying snakes into a different group that we call squamata, and then classifying crocodiles and birds into yet another group that we'll call archosaurs, we can put all of these reptiles, including birds, into a single monophyletic group or into a clade. And this makes it a little bit easier to understand their evolutionary relationships. So a lot of this is really just based on the naming of things. None of this changes patterns of relatedness. None of this changes nature. This is just changing the way that we're classifying things and naming them. Reptilia versus Aves is one classification scheme. Squamata versus Archosaurs is a different classification scheme. If you look at these phylogenetic trees, they are exactly the same. This just allows us to sort of more neatly classify these things. And this kind of challenges the Linnaean classification system a little bit, but folks are dealing with that nowadays. And so if you come across information showing how some things were classified as, let's say, reptilia at one point in time, but now they're really kind of classified into a different group, it's because of this type of. Sometimes it's a little more difficult to get away from this problem, and we see this in plants. Specifically, when we look at the earliest diverging plant lineages, the liverworts, the mosses, and the hornworts, they belong to a group that we call bryophytes, and these bryophytes are a paraphyletic group. Likewise, if we look at the lycophytes and the manilophytes, which form a group called pteridophytes, these are basically ferns or fern-like type organisms, they also form a paraphyletic group. So, for example, the bryophytes, they share a common ancestor with all of the taxa on the tree, but only these three descendants. Likewise, the pteridophytes share a common ancestor here, that is share a common ancestor with lycophytes and manilophytes, and also with the seed plants. But pteridophytes do not include seed plants. That makes them a paraphyletic group. Paraphyletic groups are often referred to as evolutionary grades. So these are points in time where there was some common ancestry that led to the descent of a whole bunch of different lineages. But there were other descendant lineages from this group that form clades that are not included within the grade. And a lot of this just has to do with how much evolutionary change do these groups share or not. And if they shared a lot of evolutionary change, then we might be inclined to regroup them together. But if they don't, as is the case with these plants, we kind of get stuck with these paraphyletic grades. Lastly, we have these polyphyletic groups, and these polyphyletic groups share traits not inherited from their most recent common ancestor. And this might sound familiar. These are traits that evolved via convergent evolution. And a classic example of this, again, are things that fly. Hummingbirds, well, any flying birds, really. Bats, which are mammals. Butterflies, which are arthropods. All three of these different things have wings, and all three of them can fly, but we know that they did not inherit flight from a common ancestor. So this makes them a so-called polyphyletic group. We can think of in the hypothetical cartoon here, maybe these are birds here in E, and these are bats here at G. And so they're grouped together by the virtue of the fact that they both fly, but they did not inherit flight from any of these common ancestors along the way. And taxonomic revisions, the process, again, by which we start renaming things, which is a messy process, but it happens because it improves our understanding and interpretation of evolutionary relationships. These taxonomic revisions can make formerly monophyletic groups, or clades, it can turn clades into polyphyletic groups. And a good example of this comes with raptors. Biologists originally thought that falcons and eagles, which share a lot of characteristics in common, shared a common ancestor, and they were grouped together in an order known as the Falconiformes. Evidence came to light to suggest that actually falcons are a lot more closely related to songbirds, and even still more closely related to woodpeckers than they are to eagles. And so this forced a taxonomic revision wherein the group Falconiformes was broken apart, and eagles were taken out of the group Falconiformes and put into a new group that was called the Acipteriformes. And so now the modern phylogeny of these birds puts eagles as an outgroup in their own separate lineage, and puts Falconiformes as a sister taxa with the passerines, or the so-called songbirds. And so these raptors are a polyphyletic group. They share quite a few characteristics in common, but they did not necessarily inherit those characteristics from a common ancestor. And that's because of convergent evolution. And lastly, this one idea comes up that sometimes taxonomic relationships simply just remain unresolved because there's not enough information to figure out which lineage is more closely related to which. And this leads to something kind of also a little bit messy called a polytomy, but this is a point in a phylogenetic tree where more than one lineage takes off from the point in time where a common ancestor is depicted. So we, for example, here have a fully resolved tree where we're pretty clear about the evolutionary relationships amongst these 5 taxa. In the second example, we see where C, D, and E, their relationship is not fully resolved, and so we see 3 lineages branching from a single point. An even more messy situation would be a completely unresolved tree called a star tree, where we might have all 5 lineages in the tree emerging from some common ancestor, but we really have no idea at what point in time any pair of them branched off from subsequent common ancestors. And this is an ongoing problem in our understanding of evolutionary relationships. However, with the advent of using a lot of genomic data, we're more and more getting to the point where these kinds of problems are starting to become solved. So that's it for this lecture. I'll see you guys again next time.